Let's begin with some general information. These organisms often originate from the mouth flora and may become pathogenic in immunocompromised hosts. They can cause periodontal and jaw abscesses, especially involving Legionella, Aggregatibacter, and Capnocytophaga. Infectious endocarditis. Bacteremia, particularly with Capnocytophaga and Cardiobacterium. They typically grow slowly and require an environment with 5 to 10% carbon dioxide. And importantly, they show no growth on MacConkey agar. Now let's look at Capnocytophaga. It is capnophilic, meaning it thrives in increased carbon dioxide. It has a fusiform shape. Clinically, it is associated with bacteremia and septicemia. Microscopically, it shows gliding motility, a characteristic movement along surfaces. Next is Bordetella pertussis, the organism that causes whooping cough. It is a gram-negative coccobacillus that requires special media for growth. The classic medium is Bordetella gengu agar, which is a potato infusion with glycerol and 20% sheep red blood cells with added penicillin. Another commonly used medium is Regan-Low agar, which contains oxoid charcoal, 10% horse blood, and cephalexin. It also has a longer shelf life than Bordetella gengu. Historically, the cough plate method was used for collection, but today it is more reliable to collect a nasopharyngeal swab and plate it directly. PCR is often used to confirm identification. On culture, Bordetella pertussis colonies require 72 to 96 hours of incubation at 35 degrees Celsius. Colonies appear pinpoint and resemble mercury droplets. The HACEK group includes a set of Fastidious Gram-negative bacteria that are part of the normal oral flora but can cause serious infections, particularly endocarditis. Let's go through each one. Aggregatibacter Haemophilus, formerly known as Haemophilus Haemophilus, is oxidase negative or only weakly positive, catalase negative, nitrate positive, and indole negative. Its colonies resemble those of Aggregatibacter species, and it is an important cause of endocarditis. Aggregatibacter actinomycetum comitans, once called Actinobacillus actinomycetum comitans, may be oxidase variable, is catalase positive, nitrate positive, and indole negative. It is strongly associated with periodontal and jaw abscesses and is commonly found in dental plaque. Microscopically, colonies may show a distinctive six-pointed star in the center. Cardiobacterium hominis is oxidase positive, catalase negative, nitrate negative, and indole positive. It is a known cause of endocarditis. Interestingly, it can give false positive gram reactions due to pleomorphism, and in culture, it often forms rosette groupings. Legionella corrodens is oxidase positive, catalase negative, nitrate positive, and indole negative. It is notable for a bleach-like odor when cultured. On agar, especially pitted agar, it produces 3 distinct zones of growth. Clinically, it is linked to periodontal disease and jaw abscesses. Kingella kingi is oxidase positive, catalase negative, nitrate negative, and indole negative. It is an important pathogen in children, where it causes septic arthritis and osteomyelitis. In culture, it produces spreading corroding colonies. Overall, the HACEK organisms share a link to oral flora and a strong clinical association with endocarditis and other oral or systemic infections. Haemophilus influenzae is a small, non-motile gram-negative rod. It requires special growth factors. The X factor, which is Haemin. The V factor, which is NAD. Both of these factors are present in red blood cells, but they are not freely available. Heat is needed to break down the red cells and release these factors, which is why chocolate agar is used for culture. A unique phenomenon with Haemophilus is satellitism. Staphylococcus aureus produces the V factor and also releases the X factor by hemolyzing blood. As a result, Haemophilus will grow in the hemolytic zone surrounding staph colonies on a blood agar plate. To observe this, You need about 5% horse or rabbit blood for hemolysis to occur. Key characteristics include: growth occurs only on chocolate agar, not on plain blood agar, unless mixed with Staphylococcus aureus. X and V discs can be placed on Mueller-Hinton agar to detect growth requirements. Haemophilus quad plates can also be used. A porphyrin test helps assess the requirement for X factor. When performing susceptibility testing, standard Kirby-Bauer zones don't apply. Instead, Haemophilus test media, HTM, should be used, with Mueller-Hinton agar supplemented and incubated in 5 to 10% carbon dioxide. It is important to perform a beta-lactamase test to determine resistance to ampicillin. Other treatment options include cefuroxime, ceftriaxone, and cefotaxime, particularly for meningitis. Clinically, Haemophilus influenzae causes several important infections. Meningitis, especially in children between 2 and 4 years old. This is usually preceded by nasopharyngeal colonization and bacteremia. Epiglottitis, typically in children up to 2 years of age. Throat cultures should not be collected since manipulation may cause airway obstruction. Blood culture is preferred. Intubation may be necessary. Pneumonia, especially in children under 5 years and adults over 60. Conjunctivitis, also called pink eye, which is highly contagious. Another species, Haemophilus ducreyi, is associated with chancroid. This causes painful genital ulcers, also called soft chancres. A Gram stain of the drainage shows tiny Gram-negative coccobacilli arranged in a school of fish pattern. Let's further review and distinguish between different Haemophilus species based on their requirements for X factor, hemin, V factor, NAD, and whether they show beta hemolysis. Haemophilus influenzae requires both the X factor and the V factor, but it does not show beta hemolysis. Haemophilus parainfluenzae requires only the V factor, not the X factor, and it does not show beta hemolysis. Haemophilus ducreyi, the cause of chancroid, requires only the X factor and does not require the V factor. It does not show beta hemolysis. Haemophilus hemolyticus requires both the X and the V factors, and, unlike H. influenzae, it does show beta hemolysis. Haemophilus parahemolyticus requires only the V factor and shows beta hemolysis. Haemophilus aphrophilus, now classified as Aggregatibacter aphrophilus, does not require either the X factor or the V factor, and it does not show beta hemolysis. So, to summarize, if an isolate requires both X and V but does not hemolyze, it's H. influenzae. If it requires both and is hemolytic, it's H. hemolyticus. If it needs only V, think of H. parainfluenzae or H. parahemolyticus, with hemolysis distinguishing them. If it needs only X, it's H. ducreyi. If it needs neither, it's H. aphrophilus. Let's continue with Legionella pneumophila. This organism causes 2 forms of disease: Legionnaires' disease, which is a severe pneumonia, and Pontiac fever, which is a much milder respiratory illness. About 3/4 of all cases are due to Legionella pneumophila serogroups 1 and 6. The bacteria are found in potable water, in faulty air conditioning vents, and in natural sources like lakes and ponds. For identification, the best specimens include sputum, bronchial washings, pleural fluid, or lung aspirates and biopsies. In the laboratory, Legionella grows on buffered charcoal yeast extract BCYE agar, with or without antibiotics. But it will not grow on chocolate agar or blood agar. For direct examination, stains such as Giemsa or Gram stain with a basic Fuchsin counterstain can be used. Now let's move to Mycoplasma and Ureaplasma. These are the smallest free-living microorganisms, measuring only 125 to 250 nanometers. They lack a cell wall and are instead bound by a triple-layered membrane. Because they lack a cell wall, they do not stain with the Gram stain, but they can be visualized using a Dianese stain. On special media containing sterols, Their colonies have a characteristic appearance where the center grows into the medium, giving the look of an inverted fried egg. Mycoplasma pneumonia causes primary atypical pneumonia, also called walking pneumonia. This condition is common in young adults and children. It can cause a positive cold agglutinin titer above 1 to 32, and sometimes a false positive RPR test. Treatment is with erythromycin or tetracycline. Mycoplasma hominis can colonize the genitourinary tract and may cause postpartum fever. Unlike other mycoplasma, it is resistant to erythromycin and is instead treated with tetracycline. Ureaplasma ureolyticum is another species that causes non-gonococcal, non-chlamydial urethritis, especially in males. It is notable for producing ureas. Treatment options include tetracycline or spectinomycin. Finally, let's discuss the Spiralaceae family. These are rigid, helically curved rods that move with a corkscrew-like motility due to polar flagella. One important member is Spirillum minor, which causes rat bite fever. It can be visualized by dark-field microscopy or with Giemsa stain. The organism has 2 to 3 spirals and bipolar polytrichous tufts of flagella. It does not grow on artificial media. This chart summarizes important fastidious gram-negative rods that require special laboratory conditions. Bordetella pertussis is a gram-negative coccobacillus. On culture, it produces pinpoint colonies that look like mercury droplets. Clinically, it causes pertussis, also known as whooping cough. The organism is best cultured using Bordetella medium, and the preferred specimen is a nasopharyngeal swab, which should be plated directly. Haemophilus species are small, non-motile gram-negative rods. Within this group, Haemophilus influenzae causes influenza-like illness, meningitis, and epiglottitis, while Haemophilus ducreyi causes chancroid, a genital ulcer disease. On gram stain, H. ducreyi often appears in a school of fish arrangement. These organisms require both X factor, hemin, and V factor, NAD, for growth, usually supplied by chocolate agar. Legionella pneumophila grows on BCYE agar or buffered charcoal yeast extract medium. It is responsible for Legionnaires' disease, a severe pneumonia. Unlike Haemophilus or Bordetella, it does not grow on routine culture media. Mycoplasma and Ureaplasma are unique because they lack a cell wall. Their colonies grow into the agar, giving them the appearance of an inverted fried egg. Mycoplasma pneumonia causes primary atypical pneumonia, also known as walking pneumonia, and is associated with a positive cold agglutinin titer. These organisms cannot be stained by gram stain but can be seen with the Dienes stain.